A very good morning to one and all. So today is the third day of our international webinar series, and we have with us our eminent speaker for the day, Dr. Abhijit Date. We have got two moderators today, Dr. Madhur Kulkarni and Dr. Digambar Ambikar. Also, we have with us our principal, Dr. Anagha Joshi, as well as our team member, Dr. Mansi Vagdarikar. So, hearty good morning to all of you. And now I hand over to Anagha ma'am.
Okay, so a very good morning to one and all. In Pune, it's a nice weather as well, Abhijit, today. And it's a pleasure and honor for me to welcome you, especially for this international webinar series, the third webinar in the row. And I welcome all the viewers and eminent senior teachers also for this particular webinar on the theme of biologics. I must put it on record and thank Abhijit from bottom of my heart for helping us coordinate and arrange this series in a sequence and basically coming us uh, narrowing down to a particular theme which is very pertinent in this particular scenario. So I welcome all of you once again and enjoy the seminar and make maximum use of this. Thank you.
Thank you, ma'am. As well as Dr. Abhijit is an adjunct faculty of Team ICP. So, it is a great pleasure, an additional pleasure for all of us. Now, I request Dr. Digambar Ambikar to kindly introduce the speaker for the day. Yeah, Dr. Digambar, please introduce. Kindly unmute. Yeah, Dr. Digambar, am I audible? Please unmute.
I think Digam.
Yeah, there is some connectivity issues.
I'm calling him just a minute.
Yeah, please. Just a matter of time. Everything will be all right. Just please bear with us. Probably there is some technical snag. That's it. Actually, from today, all our team has started going to the college, and most of 50% of them are from the working from the college, and others we are working from home right now. So probably there is some connectivity issue or something. Just let us sort it out.
Like if Dr. Digambar is having any problem, I think I'll just
Yeah,
introduce
please, ma'am.
Dr. Abhijit briefly.
Yes, ma'am.
Abhijit Date, MPharm PhD, is an assistant professor at the Department of Pharmaceutical Sciences, the Daniel K. Inouye College of Pharmacy, University of Hawaii at Hilo. He received his MPharm and PhD in pharmaceutics from prestigious Bombay College of Pharmacy, University of Mumbai, India. After graduation, he received postdoctoral training at the Chemistry of Life Processes Institute, Northwestern University, School of Pharmacy and Health Professions, Creighton University, and at the Center for Nanomedicine at Johns Hopkins University School of Medicine. He received the Johns Hopkins Center he received the Johns Hopkins Center for Nanomedicine Research Excellence Award in 2016. He also received the Hilo College of Pharmacy Student Choice Award for teaching pharmaceutical sciences for the last 3 years. He has 48 peer-reviewed publications and 4 book chapters to his credit. His research work has resulted in the filing of 4 US patents. With this very brief and very modest introduction of Dr. Abhijit, I hand over the session to Dr. Abhijit.
Thank you very much, Dr. Madhur.
All right. So thank you all for the invitation, and thank you for the kind introduction. I hope I'm greetings from Hawaii, and I'm hoping that these are very difficult times, and I'm hoping that things are slowly getting back to normal at home. But certainly technology has made things possible. I can connect with all of you all the way from Hawaii and still share some of the thoughts. So I'm glad to get this opportunity to speak with you all and share some thoughts on gene therapy. Hopefully these are in line with the current topic for this international webinar, right? So there are a few things that I just want to talk about before I really get started. So what I've tried to do, again, as you can see from the topic, it's a pretty broad topic, right? I mean, you can spend entire semester actually, or even whole academic year talking about different modalities and vectors for gene therapy, which is such an extensively researched topic, right? So what I've tried to do today is that, I've tried to give you all brief overview of some of the technologies that are already in there, and I've tried to discuss some key technologies which have been translated to the clinic, right? So this is sort of an overview. I've tried to include some details, and hopefully this will serve as a good starting point for students as well as instructors. Wherever possible, most of the cases I've included straight away the HTML link, so that if you guys want to go to those links and read those papers, I think that would be very valuable. It's a pretty big topic. I've tried my best to put together in about an hour, in about 55 or to 60 slides. Okay. All right, so just brief outline what I'm going to talk about. I'm just going to give a brief background of gene therapy. We're going to start with approved products. So I want to tell you guys that, well, this is a reality. Gene therapy is a reality. And we slowly start deconvoluting. We're going to talk about different forms of gene therapy, different modalities that we're going to use to achieve gene therapy. Last but not the least, vectors of different types, which include viral, non-viral vectors. And lastly, some limitations. This is what I'm going to talk about today. All right, so I thought this, I found this image, and I thought this is very powerful, right? I mean, we all know about gene therapy. I mean, I don't want to go into too many details, of why do we need gene therapy, but this particular image is, I think, very powerful, right? So while we have so many diseases, but there are several rare diseases which are there for which there is either no treatment or barely any treatment, right? So I think this particular slide or image talks about rare diseases and the prevalence in only US and Europe. But if you look at the global scale, I think the number is definitely higher. So for all these rare diseases, up to 7,000 or even more, most of which are of genetic origin, we really don't have any treatment. And gene therapy is literally our last and only hope, right? So this is one of the major motivations for gene therapy. And this is, I personally think that this is a very powerful slide. And if you can really make gene therapy possible or feasible, then it's going to change and transform lives of all these people around the globe, right? So definitely people have been trying to do gene therapy for decades now, right? And hopefully in the next hour or so, I'll try to give you some account of what all things we've done and where we're at currently with respect to the approved products, okay? All right. Okay, so another just more sort of summary slide, like gene therapy. If you look at this is I was trying to find out like really up-to-date figure up to 2019, but fortunately I couldn't really find a figure that talks about all the gene therapy trials so far. But if you look at this figure, you can see that almost 2 decades or more people have been looking into different types of gene therapies, right? And if I'm right, there are almost like total number of trials is around 2600, right? So you can really see people have been extensively looking at different forms of gene therapy, right? So this clearly highlights the interest as well as the potential of gene therapy for not only currently, but also for the future, okay? All right. So I was debating where should I really include this slide, right? I mean, we could sort of put it as a culmination talking about gene therapy as a reality, but I thought before I really start talking about all the basics, I wanted to tell you guys that gene therapy is a reality, right? So this is just a brief summary of some of the products. I've just tried to pick up, either the product labels or stuff like that so that you can see. And you will definitely have links for more details later on, right? So the bottom line is gene therapy is a reality. We've been looking at it for several decades now. We've definitely had our setbacks. Almost 20 years ago when people started dabbling with viral vectors. But even though we've had setbacks, continuous efforts, continuous developments in genetic engineering, in delivery technologies, and our understanding, of the genetic basis of life or diseases has definitely made gene therapy feasible. Okay, so these are just some products. Most of these are recent products that I've just included there, but you'll see there are way more products available in the market. The next slide, again, sort of a summary slide, briefly talking about some of the gene delivery or gene or cell-based gene therapy products that are currently approved across the globe. I mean, most of those are approved in the US, but there are some of these are approved in Europe or even other countries such as China or Japan. Okay. Now you'll see here, again, I don't want to go through a lot of product details, and we will definitely talk about different modalities and vectors, but you'll see two different types of gene therapies here, right? So one of them is in vivo gene therapy products, right? So the idea is that you have a product which is going to be injected into the body or administered to the patient in vivo, either locally or systemically, in different forms using different vectors, so that you can really get the gene, achieve the gene therapy. Or the other type of or form of gene therapy that you can see here is ex vivo gene therapy. So the idea here is that you want to isolate the cells from the person or from the donor. You want to modify or genetically manipulate those particular cells to achieve, in most of the cases, cancer therapy, right? And you use vectors, you use certain genes, you modulate the cells of the donor and those cells. So this is ex vivo gene therapy, right? So the idea is that you ex vivo modulate your the cells from donors, and once you modulate that, you inject those cells which contain that modified gene or modified genetic material, and you inject them into the patient, and those injected cells eventually will elicit the desired response, right? So this is like a broad form of gene therapy, ex vivo as well as in vivo gene therapy. Okay. All right. So I'm gonna try and talk about different forms of gene therapy. Again, the idea is that I'm just trying to lay out some basics so that even students can understand. I've tried to be as simple as possible so that and even during the presentation, if you have any questions, you're welcome to ask them. Okay, so there are different forms of gene therapy. We're going to talk about, I just introduced you guys to two different types, which you see here, right? Gene therapy, where you're trying to introduce genetic material with the help of vector into the, into the person, either locally or via injection. The cell therapy is typically ex vivo, whereby you are modifying the cells using genetic vectors, and right modalities, and which you will inject back into the patient. And third one, which is also a form of gene therapy, which essentially means is that I'm sure you may have heard about technologies, editing technologies such as CRISPR-Cas, which have really picked up in the last, I would say, 5 to 7 years, right? So in this case, you're not necessarily introducing genetic material into the body, either in vitro, in vivo, or ex vivo. You're actually introducing modalities which will edit the gene of interest so that you can either correct a disease or you can introduce or you can correct the defect into the genetic code, and you can achieve the therapy. Okay, so we're not really going to talk about a lot on gene editing in the interest of time, but I'm going to try and cover at least gene therapy and different modalities for that. Okay, but you will definitely find a lot of resources for CRISPR-Cas or zinc finger nucleases and some of those modalities which are available for gene editing as well. All right, so again, this is a basic explanation of what are different forms of gene therapy. I've tried to be pictorial and simple. Okay, so how can you really achieve gene therapy, right? So if you look at genetic diseases or inherited diseases such as say hemophilia, which basically are characterized by absence of clotting factors, right? So and there are several such diseases where the gene of interest is actually not available in the person, or is missing in the person, and you essentially gets that disease, right? So in those cases, you can think about the genetic sequence. If you know the gene sequence, you can figure out a vector, you can figure out a modality, and you can insert this gene which is non-functioning or either absent in the person or non-functioning. You can insert that, and once you insert that gene into the person, you can essentially correct that genetic disorder, right? So it's one form is essentially insertion or augmentation, right? Hemophilia could be a good example. The other type of gene therapy is correcting or inhibiting the faulty gene. So, for example, there are several genetic disorders which emanate from because of faulty genes. So, one example can be cystic fibrosis. You have CFTR misfunction, and definitely, if you identify those mutations in the gene, and if you want to correct them, then you can certainly identify the faulty gene. You can certainly correct those mutations by blocking that gene. And then once you block that gene, is essentially that fault is going to be repaired, and you will see restoration of the function, right? So I think that's one way in which you can think about gene therapy. The third type is using a suicidal gene, right? Or in which you're essentially using genes to activate certain proteins or certain signals inside the body or inside the cells, and that will lead to cell death or essentially correction of disease. So if you've heard of proteins such as p53, which is apoptotic protein, you can certainly encode encode the gene containing that gene which encodes that protein. You can put that into the vector, and you can introduce into the cell, and that will essentially cause cell lysis. Or the other example of suicide gene therapy is that if you people use suicide gene therapy to selectively kill cancer cells. So, for example, your nucleoside analogs such as ganciclovir, right? So, people use viruses such as herpes simplex virus thymidine kinase, which contain a specific enzyme which will activate these nucleoside prodrugs only into the cancer cells, right? So, I think that's another way in which you can kill specific cells such as cancer cells by using gene therapy. The other version of this suicidal gene therapy is that you could essentially deliver marker gene. We'll talk about the vectors and modalities in next slides, but I'm just trying to clear the approaches overall, right? And the marker gene will essentially be introduced into the cell. It will get transcribed, translated into the proteins which are specifically required for immune activation. And once these proteins are expressed, your immune cells are going to attack these cells, and then the disease cell will die. So this is somewhat similar to, I would say akin to mRNA vaccines, right? So, and I'll talk about mRNA vaccines or mRNA-based modalities in a few minutes. But the idea is that you will introduce a protein which is basically immunogenic protein from a virus or bacteria or your target disease, and then your body will essentially generate immune response to that, and which will lead to cell killing. So mRNA vaccines is a very powerful tool, and I'll again talk about that in next few slides. Okay. All right. So before we really talk about modality, just want to briefly talk about, or at least talk about transcription, translation, and genetic basis, right? So your journey from DNA to protein, once you understand that, essentially you can understand the modalities that we have for gene therapy. So I'm going to briefly discuss that, right? So I'm sure we all know that, our chromosomes, which contain, they are made up of DNA, which contains our genetic material. So a brief description of transcription process is that your DNA is transcribed using your enzymes such as RNA polymerase, which leads to formation of messenger RNA, which essentially gets translated further using different mechanisms and with the help of ribosomes, and then you start getting expression of proteins, which will act in the body and which will allow for normal cellular function, right? So I think it's a brief protein synthesis process. And if you look at the transcription and translation process more closely, you will understand that if you really want to make changes at the genetic level, if you modulate these events, say transcription or translation, essentially you can think about developing modalities for gene therapy. Okay. Again, this is a brief description. We don't want to go into genetic engineering lectures right now because, again, I am trying to give you guys an overview. But I would certainly encourage you all to read a little bit about transcription, translation. Again, whenever you're trying to revise, and then you will understand modalities even more. Okay. All right. So again, this is a brief description of, or summary of modalities for gene therapy. I know it's a pretty busy table, but I really like this table. And again, I've given you the resource. If you this is a really nice paper. Review paper that I found, and this will sort of tell you the chronological events of gene therapy. We're going to discuss at least the first few of them. We're not going to talk about restriction enzymes or gene editing tools such as zinc finger nucleases or TALENs or CRISPR-Cas9, but we will definitely talk about your antisense oligonucleotides, miRNA, siRNA, mRNA, and plasmid DNA in the next few slides. Okay, but this is just a nice summary of different modalities that we have for gene therapy, which people have been working on, and their potential mechanism of action. And obviously, in the next few slides, you'll start seeing more details. All right. So before we really talk about gene therapy, I want you guys to think about the barriers, right? I mean, it sounds very cool. I mean, Human Genome Project allowed us to definitely profoundly improved our understanding of genetic basis of life. We know that we definitely have figured out ways to manipulate things at the genetic level. It all sounds very cool. It's very, very specific, very powerful. But then, gene therapy is as cool as it is. It's also very tough. I mean, I want you to understand the differences, the barriers, and that will certainly highlight the importance of vectors as we go along further, right? So just like, I've tried to include your extracellular barriers as well as intracellular barriers. So some of these barriers are obviously common when you try to deliver drugs into the body, right? So at the end of the day, we are trying to introduce genetic material in the body. So it is going to be treated by the body as a drug. And we have to think about all considerations when it comes to delivering drugs or all the barriers to drug delivery, right? So a couple of things which are even more specific when it comes to nucleic acid therapeutics or gene therapy. You definitely need to think about extracellular barriers such as nucleases, right? So our body is your nucleases, which can selectively cleave off your nucleic acid materials are present everywhere. So I think even if you figure out a solid genetic sequence or gene or siRNA, any of the modalities that I briefly talked about earlier, even if you figure that out, a very solid sequence that you see, is very potent, the moment you inject that in the body, your nucleases are going to destroy them or destruct them immediately, right? So your nucleic acid modalities are inherently unstable when you inject them into the body. So I think that's one major barrier you need to think about, the stability, right? Our body's clearance mechanisms are also going to be there. So the moment you inject these materials into the body, you're reticular endothelial systems. Your MPA is essentially going to recognize that. They're going to immediately start working on clearing these out, right? So if you inject only naked nucleic acid therapeutics, people have found out using studies that the half-life is barely minutes, right? So that's how powerful our body's clearance mechanism is, right? So I think that's one barrier that you need to think about: enzymatic degradation using nuclease. The second barrier is your body's immune system. The clearance mechanisms of the body, a lot of them will be cleared immediately, literally within minutes, if you don't take proper precautions. And you'll see a lot of the genetic material or modalities landed in like your Kupffer cells in the liver, and eventually excreted by the urine. Okay. Another important factor or barrier when it comes to delivery of genetic materials is that tissue penetration. I mean, these are nucleic acid materials, right? So, very hydrophilic, very highly anionic materials, right? So these physiochemical properties, so think about your nucleic acid therapeutics as BCS class 3 drugs, right? Which are poorly permeable. We all know that the availability of these sort of materials to the body is very minimal because of their permeability issues. So these same issues are going to be there when it comes to different types of nucleic acid therapeutics. So because of their highly anionic nature, highly hydrophilic nature, your cells are unable, even if you somehow figure out a way to deliver your siRNA or other nucleic acid therapeutics, your cells are not going to be taking them up really well, right? So that barrier is definitely going to be there. The next in line is your intracellular barriers, right? So, and this is where, your gene therapy or nucleic acid materials is largely different from your conventional drug therapies. I'm sure you all know, when we talk about drugs, a lot of the times, most of the times, the target of the drugs actually lies extracellularly. It may be a surface receptor, a protein that's already expressed. Most of the times, it's either, most of the times it's on the surface, right? So your drug basically has to reach to the surface of the cell and essentially act so that it can elicit its action. On the other hand, your nucleic acid therapeutics have to be typically delivered intracellularly, and that definitely adds one more barrier, right? So depending upon the modality, again, which we'll discuss in some time, you would need your nucleic acid material at least in the cytoplasm, or in many cases, you may need it actually into the nucleus. And it sounds very it sounds very cool to discuss, but it's a very tough journey for when you're trying to deliver something external, right? So some of the things that you need to think about whenever it comes to intracellular delivery, one of the major barriers for gene therapy is the endosomal mechanisms in the body, right? So your cells are capable of treating foreign substances by capturing them into endosomes. Endosomes typically tend to be acidic in nature, and this is how your cells will basically kill foreign substances such as bacteria or viruses, right? They'll change the acidic nature as well as the lysozymes. All these enzymes intracellularly will degrade your foreign substances as well as your exogenously delivered nucleic acid material. So whenever you're talking about delivery to achieve the gene therapy, you need to think about intracellular barriers also very carefully. And again, this is just all the way up to cytoplasm. If you're looking at delivering plasmid DNA, again, we'll talk about that in a few minutes. If you're looking at nucleic acid materials which need to be delivered to the nucleus to elicit its effect, then you're talking about transport from cytoplasm to nucleus, and that itself is additional barrier, right? So you need to think about all these barriers whenever you're trying to think about gene therapy, and that's why it's taken a really long time. Although we've been certainly successful, but all these barriers are very complicated, and they all act in tandem, and you need to really figure out a way to overcome all of these if you really want to achieve successful gene therapy. So again, I wanted to give this background a little bit earlier because when I talk about modalities, I'm also going to talk about some modifications so that we can overcome this barrier. So that's why I thought it would be a good idea to talk about barriers first before talking about modalities. Okay. All right, so let's talk about different modalities, right? So, I told you guys, I'm going to go back a few slides and go to that, transcription translation side, right? So one of the ways in which you can essentially manipulate or get the gene therapy is by simply inserting the gene itself into the nucleus, right? So I think that's one way. Even before transcription, you essentially figure out a way to deliver the gene directly into the nucleus, and one of the modality which allows you to do that is your plasmid DNA, right? So here, the idea is that you know the sequence of the gene already. Your Human Genome Project already has enabled that. You know the sequence of the gene that you want to deliver, and bacteria have been using this large circular plasmids for a long time. They're highly conserved. They're pretty big. They are capable of carrying large genes, right? So people have been looking into plasmid, DNA for a long time to deliver genes, either into cells, in vitro, ex vivo, or in vivo for different times. So plasmid DNA is one modality that was, early on. People started working on plasmid DNA. The idea is that, well, you have this bacteria-derived circular DNA molecules. They're pretty bulky, as you can see. They can be as small as 1,000 base pairs to all the way 200,000 base pairs. So it's a pretty bulky molecule which can certainly accommodate the gene. Again, these are of bacterial origin, right? So you can see that if you look at the plasmid design, you can see that there are some aspects. There are some components that are retained from the bacteria, such as your antibiotic-resistant gene. That's there from the bacteria. You can see in the figure that you have a trans gene of interest, right? So you can manipulate the bacterial plasmids in such a way that you can insert your gene of interest into this plasmid. You also have to think about some other aspects when you design a plasmid. You want your plasmid to be promoted into different cells, right? Because you're using bacteria to essentially express this plasmid on a really large scale, which contains your therapeutic gene, right? So you have to, whenever you're talking about this modality, you need to think about designing plasmid in such a way that you will have a stable insertion of your gene. You will be able to create, replicate plasmid DNA using, simple fermentation with the help of E. coli, and you can produce this in a large scale. And eventually, if you figure out a way to deliver this plasmid DNA into the cells, into the nucleus of the cells to be specific, you will be able to treat the cells, insert the gene into that particular cell, right? So I think this is one of the early modalities that people started working on. I'm going to focus a lot more on RNA, and the reason for that is that one of the reasons for a lot of focus on RNA molecules is your plasmid DNA has to be delivered to the nucleus, right? So even the journey from cytoplasm to nucleus is really hard, right? So if you can avoid that, that's something people realize. I mean, people did try and experiment with plasmid DNA for a long time. We definitely don't have any commercial products based on plasmid DNA. People have looked at plasmid DNA-based vaccines and several types of modalities, but because the barriers are even more, definitely they haven't been as successful, and that's why people started looking at RNA-based therapeutics. So I think next few slides we're going to talk about more RNA-based modalities. Okay. All right. So one of the early on modality, which is much shorter compared to plasmid DNA. You saw plasmid DNAs are pretty bulky, right? One of the early modalities that people started looking at, the idea was that, well, preferably, if possible, we don't want to go all the way into the nucleus. We need to figure out a way to modulate, your genetic sequencing in the cytoplasm itself. And if we can achieve that, essentially, we can still get the gene therapy. So the idea, again, is that you're not looking at, most of the cases, you're not looking at getting into the nucleus, but you're mostly looking at cytoplasm modulation and essentially which will elicit the gene therapy. So one of the early ons is antisense oligonucleotides, which are essentially single-stranded synthetic RNA or DNA molecules. Again, these are much shorter. You can see these are these can be anywhere between 12 to 25 nucleotides in length, right? So it's a very short fragment, nucleotide fragment. The idea here is that, again, the majority of the focus of antisense oligonucleotides is in cytoplasm. The idea is that, well, your DNA, with the help of your DNA, is going to get transcribed into mRNA, and mRNA is going to get translated into the proteins, right? So if you figure out a way to mess up with mRNA in different ways in the cytoplasm, and if you either block the mRNA or you figure out a way to degrade the mRNA so that it doesn't get translated to the defective protein or protein of interest, then essentially you can achieve the gene therapy, right? So the idea of antisense oligonucleotides is that they've been designed in such a way that they will bind to a particular sequence in the mRNA, and once they bind to this mRNA sequence, essentially your translation process of the mRNA is going to get messed up, right, or get distorted. And once that happens, essentially you're not going to get the faulty protein that you're trying to get rid of, and which will eventually lead to therapeutic effect. So again, it's a simple idea of much shorter strand of oligonucleotide strands. Mostly focuses on cytoplasmic delivery, as you can see here in the slide. There are several ways in which antisense oligonucleotides act once they bind to the mRNA sequence. You could get a steric blocking, right? So idea is that this antisense will sterically block the mRNA translation, which will lead to protein, or they can actually activate an enzyme called as RNase H, which can essentially cleave the DNA, which will lead to a failure of translation of the protein. There are some approaches where people are trying to look at delivering antisense all the way into the nucleus. Again, it's much tougher compared to cytoplasm, but by design they can be also designed in such a way that you can try and get to the nucleus even before the mRNA gets released into the cytoplasm, and you can essentially block the protein translation. Again, if you just think about transcription translation process, and if you know the different ways to mess up with translation process, you will understand how different modalities for gene therapy work. Okay. All right. So I mentioned to you guys that inherently your nucleic acid therapeutics are not stable at all, right? So it sounds very cool that you have complementary antisense, it blocks the mRNA. Theoretically or even in vitro, it works out great. But then when it comes to actually in vivo, the stability of the nucleic acid, this is a major issue, right? So, over the years, people have looked at this aspect very significantly. And you can see that antisense oligonucleotides, as well as a lot of other nucleic acid therapeutics, which are currently available in the clinic, are modified nucleic acid. So you can see different levels of modification. One of the modifications that you see here is you replace the oxygen with sulfur, so you have phosphorothioate, or you can have methyl modalities, or you can ameliorate your phosphorus phosphate in there, and you can get first generation. People went even further. People started looking at modifying the sugar part of your antisense. You have methyl, you have methoxy, which clearly impart much greater stability from nucleases, as well as it significantly improves the half-life of the molecules. Okay. The third generation, where you can see that the classic DNA RNA-based sequences is totally modified, right? The one on the right-hand side, most right-hand side, you see is a phosphorothioate, phosphoramidate morpholino oligo, right? So here the sequence is, it's no longer your deoxyribose sugar, essentially your morpholino molecule, which sort of mimics your nucleotide sequence, and you have a highly modified nucleotide, which eventually can elicit genetic therapy or gene therapy. So again, chemical modifications are highly prevalent in most of the currently approved nucleic acid material-based products. Okay, these are some of the modifications that are routinely performed on antisense as well as some of the other nucleotide molecules. I just want to give you guys an example of. Sorry, I'm just want to check how am I doing on time. Okay, all right. Yeah, brevity is not my strong suit, so I'm going to try and make sure that I still deliver all the material as well as try and stick to the time, so please bear with me. Okay. All right, so I want to briefly talk to you guys about one of the recently approved antisense oligonucleotide. It's almost approved in 2019. Again, the idea here, I'm not going to go into a lot of details. The idea here is that this is sort of magnified view of modified nucleic acid therapeutics. So you can see that the currently approved modified ASO is structurally very different than your standard DNA or RNA molecule, right? So I think that's what that's the point I'm trying to get at. I mean, this is used for treating muscle atrophy, which is genetically linked muscle atrophy. Okay, another just a quick summary of nucleus ASO development timeline. So you can see that people started looking at it for almost 3 decades. There have been several iterations, but we do have several antisense oligonucleotide-based products approved. You can see first one was approved almost in late 90s, and that was actually for a local delivery. But now we have antisense oligonucleotides for IV delivery or subcutaneous delivery as well. All right. The next one in line is small interfering RNA, which is, again, siRNA. I'm sure you all have heard of it. It's a very powerful tool. It's just fun fact, siRNA, to my knowledge, is one of the discovery that received Nobel Prize in shortest time. I mean, if I'm right, within like 8 to 9 years after discovery, it received Nobel Prize. So I think this is a Nobel Prize winning technology, which has turned out to be very powerful, gene editing tool. And we do have a marketed product based on siRNA right now. We're going to talk about that after some time. All right, so again, these are, again, see the difference. These are double-stranded RNA molecules. Again, shorter molecules. The idea is that you wanna mess up the translation process, right? So, the figure here, you can see you have siRNA molecule that can be endogenously generated, or if you succeed in delivering siRNA molecule, what happens is that it's a double-stranded molecule. The cells machinery basically recognizes this molecule. The Argonaute proteins and RISC complex, they basically bind to this molecule. They separate this molecule, or you can say that it's unwinded into single strand, the siRNA. That strand will basically bind to the complementary sequence from mRNA, and once it binds there, you could essentially get degradation of mRNA, which will again prevent the translation process. So again, the idea is the same for most of the remaining modalities that mess up with mRNA translation. Is that you figure out in different ways, you figure out ways to block the mRNA translation process. Okay. Again, here you can see, like ASO, there are definitely chemical modifications that are routinely done so that you can increase the stability, pharmacokinetics, and all those properties of these molecules. One of the simple ones is 2-O-methyl, which we talked about earlier. And I'm going to actually give you guys an example of a very recently approved therapeutic siRNA molecule. So here you can see some modification. Again, I've tried to put together a structure that I found. You can see, again, this is a double-stranded, right? It's a double-stranded molecule. You have sense, antisense strand. But if you look on the left side, look at the modifications here, right? So you have O-methyl modifications, you have phosphorothioate modification, you have even fluorine molecules on certain, on certain nucleotides, and all together, along with when you attach a very bulky molecules, lipid molecules, as well as targeting sequence such as N-acetyl galactosamine, you actually, after several iterations, Alnylam has figured out a therapeutic siRNA molecule which doesn't really require any vector. So this is a very, very awesome development in the field of self-delivering siRNA or nucleic acid therapeutics. You can see the chemical structure and design here. Okay. All right. So again, this is a brief summary of Givosiran, which is again recently approved. It's a self-delivering siRNA molecule structure you already saw earlier. This is delivered subcutaneously at a very high dose, and people have figured out with the help of clinical trials. I'm not going to read out the slide because you guys can later on read that as well. The whole idea is that this basically works. It shows dose proportionality all the way from 0.35 to 2.5, like a classic drug, right? So I think that's a very powerful without really any other vector. It's a self-delivering siRNA molecule, and it's just recently approved. It's been recently approved by the FDA. So that's what I want to try and tell you. One of the overview of siRNA. Okay, the next one in line is again, again the modalities which allow for RNA, your silencing mechanisms, right? RNA silencing mechanisms. So microRNAs or mRNAs, to be specific, are again, about short nucleotide structures, right? So these are non-coding RNA molecules, so they don't necessarily code, but they code for anything. But they mess up, they modulate their functions by messing up with the mRNA translation. So they are similar to siRNA, but again, structurally they are very different. So if you look at the representation here, you can see that your mRNA molecule is like a hairpin, right? So whereas your siRNA molecule is just like two double strands of right sequence. So I think that's one major difference. You do have a lot of endogenous mRNA molecules. People have identified almost like almost 600 or 700 microRNAs, which have different types of functions in the body. Okay. Now, if you wanted to use these molecules as a modality, there are a couple of ways in which you can go about this. So I'll first tell you how RNAi is mediated using miRNA. The idea is similar to your siRNA. You have this miRNA, which you can endogenously derive, or you can deliver exogenously, right? Your Ergo as well as RISC again has a role here. This particular complex will separate the mRNA molecule, make it single-stranded, and that single strand will partially bind to the mRNA complementary sequence. Again, the mechanism is sort of similar, right? And once it binds, you can either get RNA fragmentation or you can get steric blockade, which can eventually lead to prevent the mRNA translation. Now, like over the years, people have realized that you need microRNAs for normal function, as well as there are certain microRNAs which get messed up in particular disease, right? So if you really want to use mRNA as a modality, then you can actually either go for therapeutic mRNA or mRNA mimics. So the idea is that you understand the mRNA sequence, which is required for maintenance of normal function. And if that is messed up, then you exogenously deliver that synthetic mRNA. Or if you find out that in particular disease, for example, in cancer, there are a lot of microRNAs which get mutated or the sequence is messed up, then you figure out a way to block those mRNA signals by creating a complementary sequence, something like antisense oligonucleotide, and use that sequence so that you can modulate the mRNA-based gene therapy. So you can either have antimirs, which are similar to antisense oligonucleotides, or you can have mRNA mimics. So this is one modality which allows you for genetic manipulation. All right, mRNA. Again, this is a relatively, people have been working with mRNAs for a long time, but mRNA has received attention as a modality mainly for vaccine purposes. So I wish I had I've been trying to put together so that you get an overview. So I'm just going to try and briefly describe. So, mRNA is, it's a messenger RNA. It is responsible, eventually gets translated into the proteins, right? So if you figure out an mRNA sequence, if you succeed in delivering mRNA, you still need to get to the cytoplasm, and eventually you'll get the protein translation. Now, how do people use mRNA for vaccination? I think that's very interesting. There are several clinical trials right now on mRNA-based vaccines. So the idea is that, now that we know our human genome, if you know the infectious disease, the virus or bacteria, if you know their genetic sequence of that virus, you can find out the immunogens for that, mostly for viruses. You can find out the immunogen, and you can actually create a sequence that encodes for the immunogen, and you insert that sequence into mRNA sequence, which can be delivered into the cell. So the idea is that, well, you have this mRNA which contains a sequence for an immunogen, which will eventually be translated inside your cell. And if you succeed in mRNA delivery, that genetic sequence which encodes for a particular immunogen is going to get translated into the protein or that immunogen, and your cells your immune system is going to start reacting to that immunogen, and it's going to start producing antibodies to that particular sequence. So it's a very interesting way. It's a very powerful way of generating immunity. And Moderna, like I'm sure you guys have read about COVID-19. Like Moderna Therapeutics is currently conducting a phase 1 clinical trials on mRNA, their proprietary mRNA vaccines for COVID, as well as a lot of other very infectious viral diseases. Okay. On the left-hand side, I've tried to briefly include modification. So again, a lot of these nucleic acid bases are modified. You get different methylations. I've just tried to briefly summarize that so that you can understand. But then the idea is that once you get this, you will be able to stably deliver this modality into the cell. All right, different constructs of mRNA. Again, I'll briefly go through that. So you can have this immunogenic construct that I described earlier, right? So you can have immunogenic construct built into your mRNA. You figure out a way to deliver mRNA into the cell. Your mRNA construct will get translated into immunogen, and you're going to get immune response. The other version of that is that you can actually use self-replicating viruses, genetic code from that, and build it into your mRNA. And once and then you can create something called as self-amplifiable mRNAs. And once you deliver these, essentially your mRNA copies will replicate inside the cell, and you'll get even more immune response. So it's there are so many advances. I wish even I had more time to talk about that, but there are several interesting versions that you can think of. Okay. All right. So again, this is a brief description of different clinical trials on mRNA vaccines. This is a highly sought after field for a lot of infectious disease, including COVID-19. All right, so brief discussion. We've talked about different modalities. Again, we're not even touching gene editing tools right now. We now know the barriers. We now know the modalities in which we can get genetic manipulation. Then how do we figure out a way to deliver them, right? So until we came across the self-delivering siRNAs, people have been looking at vectors to deliver these effectively, right? So you need to think about requirements for vectors. So there are several things. I've just tried to list some of them, right? You need your vector to get your genetic material to the target, so either cytoplasm or into the nucleus. If you're looking at DNA, you want significant transfection or transduction, depending upon the vector of the cell, so that you can really see the genetic manipulation that you're looking for, right? You need to have, most of the times you require long-term expression. Otherwise, you may succeed, but the effect may not really last that long. Or in some cases, you may actually desire only short-term expression. You don't want stable gene transfer. So your vector should allow you to do that. Target tissues. So ideally, vectors should allow you to get to the target tissue so that you can minimize the off-target effects. That's very important. Vector should be innocuous, right? So harmless to the patient, non-immunogenic. So these are classic requirements of your vector. It should be capable of handling a variety of therapeutic genes. So I think that's another important thing, consideration. You certainly need to be able to get dose-response relationship. As I showed you earlier, and you'll see in next slides, that you need to have a solid dose response even when it comes to delivering genetic material. And your delivery vector has to be something which is familiar and acceptable to the medical community as well as patients, right? So you want something that is acceptable, only then it can really be translated. So these are some requirements. It is very difficult to find safe and effective vectors. So I'm going to try and describe only some of them today, okay? All right, so when it comes to vectors, there are different ways in which you can vectorize or you can deliver genes. Like you can have non-viral or chemical vectors, such as lipids and polymers. You can have viral vectors. We're going to mostly focus on chemical or non-viral vectors today, but you do have viruses which have been used for a long time to deliver genes, and very early methods were physical manipulation of cells to deliver DNA or other materials such as electroporation, microinjection, or sonoporation. So again, this is broad description. If you look at the non-viral and viral gene vectors, you will see, I saw this really nice figure. You can see that, well, because viruses are very capable of delivering material into the humans, your trials on viral vectors as well as patents or publications are still sort of higher. Okay. All right, so barriers, when it comes to non-viral vectors, we have already talked about most of the barriers. I'm not going to talk about that, especially the extracellular or intracellular. But one of the main barriers when it comes to non-viral vectors is, independent of the material, is that we want to be able to create a stable complex of your vector and your nucleic acid material, which can remain stable during the storage as well as even after you deliver into the body, right? So I think that definitely adds another barrier when it comes to using non-viral vectors for gene therapy, okay? All right, so again, I'm not going to go into details, too much of details. You have different modalities. So when it comes to non-viral vectors, people have been using nanotechnology for a long time. You can have different constructs. You can have polymers. You can have inorganic materials, gold nanoparticles, silicon. You can have inorganic constructs, or you can have lipid vesicles. Could be lipoplexes or lipopolyplexes, or endogenously derived lipid vesicles such as exosomes, right? So there are several modalities that are available. Most of the discussion today is going to be on lipid nanoparticles or lipid-based vehicles for nucleic acid therapeutics. Okay. Before I really go into that, I really briefly want to describe this very solid clinical trial. I mean, another non-viral vector effort that was done by Mark Davis at MIT. It's a very powerful cyclodextrin-based vector that was generated. It created a lot of interest in the field almost for like 15 years. And, you can quickly see the design. Mark Davis is an awesome chemical engineer from Caltech. He figured out a way to polymerize cyclodextrin, add functionalities, and he even created a targeted siRNA non-viral vector. So the idea is that you have cyclodextrin, you have right modalities, you have targeted molecules such as transferrin, and you mix them all together. It's a very interesting self-assembly process. You mix them all together and it forms a really nice stable siRNA formulation. And this created so much interest in the field. And they did manage to show first evidence of pretty early evidence of RNAi, systemically delivered RNAi into the humans. Unfortunately, this particular molecule, although it showed a lot of initial promise, it did not really succeed in clinical trials. So I think I just want to give you guys some flavor of other than lipid vehicles that actually went all the way up to clinical trials, but didn't really succeed. Our focus today is going to be on gene delivery vectors. Okay, I did realize I'm going to try and give you guys an understanding of gene delivery vectors. So again, the idea is that lipid nanoparticles, people have been looking into lipid vehicles for a long time. We have several lipid nanoparticle-based approved products for drugs. The idea, again, that can we extend that, can we make modifications, and can we extend that to delivering nucleic acid therapeutics? So I'm going to take an example of the recently approved, FDA-approved product on Patro. So this is, again, lipid nanoparticle. You can see the components here. You have this cationic lipid, you have DSPC, and you have pegylated lipid. So literally these three components, and you have a modified siRNA. And together, we are able to create a lipid nanoparticle construct. And this is a very powerful example of how lipid-based non-viral vectors have succeeded, and it was approved in 2018. This is actually meant for intravenous injection. So this is another very interesting angle for that. The idea, how do we really get delivery of these lipid nanoparticles and get the siRNA delivery, right? So I'm going to go through some mechanisms of how do you get siRNA delivery in vivo after intravenous injection. So the way this lipid, the cationic lipid, and we'll come to that a little bit more, is designed is that it definitely allows you to bind to siRNA. It compacts the siRNA, it entraps the siRNA, but it also allows you to bind to the apolipoprotein or lipoproteins in the body which eventually get transferred or directed to the liver, and your liver cells take up that. So it's a very sort of passive, I wouldn't say exactly active targeting mechanism. The lipid structure obviously allows for apolipoprotein transport, and once it goes there, your liver cells, your stellate cells take up that, it goes into the cells, it fuses with endosomes in the body. Your cationic lipid basically changes the conformation, messes up and lyses endosomal membrane, and your therapeutic siRNA is released into the body, and you get siRNA silencing, siRNA mediated gene silencing. It's a very, very powerful example, already commercialized since 2018. Okay, I'm going to try and talk a little bit about the construct again, right? So the idea is, the idea is that it contains three components, right? You have cationic lipid, right? You have neutral lipid, DSPC, and you have pegylated lipid. So cationic lipid is the heart of your lipid nanoparticles for genetic material delivery or nucleic acid therapeutics. So the idea is that your cationic lipids will bind to the anionic, highly anionic nucleic acid molecules, and they will form a complex, and which will definitely allow you for better delivery as well as better stability, so on and so forth. You can come up with different designs when it comes to cationic lipids. Again, you can see three different parts. You have lipid chain, as I've highlighted here. You have a linker molecule, and you have a head group, which is quaternary ammonium or different types of amines or primary amines, secondary amines, so on and so forth. Right. So very, people have been working on cationic lipids for such a long time for nucleic acid therapeutic delivery. Right. So there are several versions that have evolved. One of the very important design consideration when it comes to lipids is PK of the lipid, and I'll come to that why it is important. Okay. You can see here you have different types of lipids that you can see. You have classic oleic acid-based lipids with different head groups that you can see here in group 2. You can have saturated fatty acid-based cationic lipids, which have mostly quaternary ammonium head group that you can see here. And the ones that actually showed in vivo therapeutic potential or delivery potential is this linoleic acid-based cationic lipids, which actually don't contain quaternary ammonium molecules. So I think these are the ones, the DC-lin, the first one in this list is one that is used in commercially used siRNA products. So you can see that part of the reason why it binds to apolipoprotein because it contains polyunsaturated fatty acid. Okay, that is where you are able to see that sort of selective transport and delivery to the liver. Okay. All right, so there are several aspects when it comes to delivering lipids. You have different structures that you can see, right? You have different head group structures. You have ether lipids, you have ester lipids, and that itself can significantly impact your siRNA silencing. You can see that the MC3 lipid, which you can see here, is very powerful in silencing at a much lower dose of siRNA. You can see the dose, even as low as 0.1 mg per kg, can silence the siRNA in preclinical studies. So very, very important to think about the structure. The pKa is very important. People realize that, one of the important reasons why we have PK, we want PK to be around 6 to 6.5, as you can see here. People try to design synthesize lipids with different pKas. What you want is that you want your lipid to be cationic enough so that you can bind to your siRNA. But the moment you inject into the body, you don't want lipid to be highly cationic in nature, because if that's the case, you're gonna get much higher cytotoxicity. Though whenever you have pK somewhere around 6.5, what happens is that if you formulate things right, it can bind to siRNA really well. If you use acidic siRNA containing buffer for formulation, the moment you inject in the body, it definitely, because of the PK change in the pH, obviously adopts more neutral conformation, so it doesn't necessarily mess up with leads to toxicity. But the moment your lipid is taken inside your endosomes, your endosome pH is acidic, again you get charge reversal, the lipid becomes highly cationic, and which will allow for disturbance of your endosome. So I think this is how your siRNA delivery, your PK of the lipid is very, very important. Okay. You have to think about other formulation aspects, such as your positive to negative ratio, such as N to P ratio or siRNA lipid ratio. You can see in this here that all those, your lipid ratio, lipid content, both of them can impact your siRNA delivery, which is another important consideration. And all right, so I want to ask organizers, I'm sorry, actually, I totally, misunderstood the time frame that I'm going to take. So I want to make sure, is it okay to continue? Or, I mean, I'm sorry about that. I mean, I thought I was going to be able to.
Dr. Kannur, you have to unmute yourself. Dr. Kannur, can you please unmute yourself?
Yeah. So am I audible?
Yes.
Yeah. So no problem. You please continue, sir. No problem at all. We are
Okay,
enjoying
right.
every bit out of it. Great.
Yeah, again, I mean, I know I've been struggling to cover this huge topic, and I think as much as I thought I would be able to do it in time, I clearly don't think so. But I do want to cover it, right? So I think it doesn't
No issues.
go well for
Please
me, I guess,
continue.
in terms of time.
It's fantastic going.
Okay, all right. Okay, sorry about that again, but I do want to try my best to deliver these things. All right. Okay, so other important point when it comes to lipids is pegylated lipids. I mean, we know pegylated lipids are used, for a long time in liposomes to allow for longer circulation. And while we certainly need pegylated lipids in lipid nanoformulation so that you can maintain the stability during manufacturing, during storage, we also know that if too much pegylation is also not very good when it for intracellular uptake, right? So I think pegylated lipids have to be critically designed when it comes to siRNA delivery. If you increase the pegylation, you may definitely get longer circulation, but you will actually inhibit or affect the cell uptake because neutral charge is not going to allow you to uptake the lipid nanoparticles. So what people have designed is that you have these shorter pegylated lipids, which essentially stay with the formulation. After you inject the formulation, your plasma proteins will actually extract this PG lipid, and then it will allow for your siRNA construct containing cationic lipid to deliver better or interact better with the cells so that you can get better uptake. So it's a very simple but very interesting design or change that people have made into currently marketed lipid nanoformulations. Okay, how do you make these? There are several ways. I mean, I don't want to go into a lot of details, such as your classic ways, such as people started with lipid film hydration. People have looked at ethanol injection, which has turned out to be really good when it comes to incorporating siRNAs. Earlier attempts were definitely with standard techniques such as extrusion, but like currently the hot topic is microfluidics, right? So the idea is that, again, there is a lot of charge assemblies here, right? So you figure out microfluidics is a very powerful way of rapidly mixing two different things in a very short period of time to get very defined constructs, right? So the idea here is that you have your acidic buffer containing siRNA, you have your lipid molecules of different sort, you mix them in two different streams in a very controlled as well as very rapid manner using microfluidics, and you can control this process and modulate in such a way so that you can get packaging of siRNA, you can construction of lipid nanoparticles, and formation of stable lipid nanoconstructs. So Precision Nanosystems is one of the leader in microfluidics when it comes to lipid nanoparticles. But again, that's a different topic. I just want to give you guys an idea of what's the current version or what's the current technique for making lipid nanoparticles. Okay, again, this is just a blown up design. The idea is that you have this very two different mixing modules. You have this microfluidic design here. You can see that the design is the design is such that it allows for intimate and rapid mixing of two phases in a very short period of time, and which will eventually lead to formation of nanoparticles. Okay, again, another mechanics of lipid nanoparticle formation. You essentially have formation of once you start mixing these together in the microfluidic device, your siRNA and cationic lipid come together first. As you go on adding more and more water-containing, buffer-containing phase, your other molecules basically start self-organizing around your siRNA and cationic lipid complex, and you get this sort of core, a very solid core shell-like structure when it comes to these lipid nanoparticles. Okay. So if you look at the TEM here, which is very powerful, very simple yet very powerful. Again, look at the TEM data here, right? Your lipid nanoparticles, when you don't have any siRNA in it, you can see that the core of the lipid nanoparticles is not as electron dense because your cationic lipid is not really interacting with anything. But the moment you add siRNA in there, you can see on the right-hand side in TEM that the core of the lipid nanoparticle is quite electron dense because you get this complex formation of siRNA and lipid molecule inside the lipid nanoparticle. So it's a very interesting design. People have tried looking at molecular dynamics. You can see the evidence here, and you also have the cryo-TEM evidence here. Okay. All right. So people have, this is an approved product, right? So they did manage to see get therapeutic effect in vivo in patients, right? This is the data of phase 1 clinical trial. You can see here especially on the right-hand side, when you have patisiran, which is the approved molecule, you really see a very nice dose-dependent silencing or knockdown of siRNA. And you can get that knockdown even as fairly low at 0.3 mg per kg of siRNA, which is pretty cool. All right, so the next version is that, because I wanted to touch base a little bit on mRNA vaccines containing lipid nanoparticles. The next design is that Moderna design, right? So I wanted to talk about that a little bit. Moderna obviously has been looking at lipid nanoparticles for mRNA vaccine design, right? So for vaccines, we need to give intramuscularly most of the times, right? So when they started looking at MC3, which is the lipid, commercially used lipid for Onpretto, they started realize that you actually the lipid really sticks around in the muscle or in the spleen or the liver much longer, which actually leads to more side effects, lipid-related side effects. So I think these guys, again, Moderna has their own biodegradable lipid platform, which I wanted to briefly talk about. So, the hallmarks sort of remain the same. You still want cationic lipid. You want to build lipid nanoparticle formulation, which can condense or which can encapsulate the oligos or mRNA therapeutics. But the design here is bit different than the MC3 lipid, which is commercially used. So if you see here, you don't have that unsaturation, but you actually have much more degradable lipids, which they actually generate using Michael addition or some of the other mechanisms. So you have lipids with pK similar to MC3, but the backbone is very different, and they have more degradable moieties in there, right? So the idea is that if you have more degradable moieties, the lipid won't stick around in the body for that long, and that will limit the lipid-related reactions that patient is getting because you're changing the site, right? So you can clearly see when they came up with their own design, you can clearly see that compared to MC3, these lipids are quickly degraded into either in the muscles or liver and the spleen, and they clearly have much better tolerability because of that. Again, this particular slide is just an example of how their new lipids were better compared to MC3. And you can see the histology slides here of the muscle and the skin, and you can clearly see that the lipid H, which is the newly designed lipid in Moderna lipid nanoparticles, it's much more tolerable compared to MC3. So I think that's another interesting change. This is a very brief introduction. This is something very new that I really liked, and I just briefly wanted to chat about that. People are also looking at, people have synthesized cholesterol-based lipids in the past, cationic lipids in the past. This group from Oregon, they figured out, well, can we actually figure out a way to change cholesterol, replace cholesterol? So you have so many pentacyclic proterpenoids, like your vitamins as well as phytosterols that we have. And I think in particular, I was pretty intrigued by the fact that some of the phytosterols, when you include them in lipid nanoparticles, can actually significantly improve your lipid, transfection or delivery of nucleic acid therapeutics. So I think that would be another direction in which one can start looking at that, how you can use even phytosterols to improve the lipid delivery or genetic material delivery. Okay. Again, the group has looked at different aspects of that. Again, there are some cationic versions of that, and you can see different levels of transfection when you make changes in the backbone or when you add some additional linkers to the molecule. Okay. So again, this is somewhat towards the future. I'm almost towards the end of the lecture. I want to briefly tell you guys about the viral vectors, because again, this is such a huge topic. I've really tried my best to give you guys an overview. Viruses, people have been using viruses for a really long time, because one of the major reasons for using viruses as vectors, because they are very efficient in getting into the human body, or even in some case integrating into your genome, right? So people figured out that, well, we can use viruses, we can modify them so that we limit, they definitely don't become immunogenic or they don't cause disease, but then we can package our genetic material or all the modalities that we described earlier into the viruses and essentially use them as a vectors for gene therapy. And people have been looking at that for a long time. We did hit a setback, I think in 20, 30 years ago when it comes to viral vectors, because they can show unexpected effects. And certainly enthusiasm was curbed at the time, but we do have viral vectors which are used for ex vivo gene therapy. So lentiviral vectors, as you can see here in the table here, they are mostly used currently for ex vivo gene therapies. And adeno-associated viral vectors, or AAV, are the currently ones which are used in clinical products. Okay, I'm gonna briefly tell you the issues with that. I mean, we definitely have several issues with viral vectors. They are very powerful, but they can have immunogenicity issues, right? So you can have, you can essentially have neutralizing antibodies for the viruses, which will inactivate them. Even after a virus manages to get into the cell, you can have your cells can figure out a way for proteasomal degradation of capsid proteins of the viruses, and they can inactivate them. So even for viral vectors, although powerful, they do have some limitation when it comes to their use as a vector. But what I want to leave you with is we do have adeno-associated viral vectors which have been commercially made available into products. So we have 2 products currently based on AAV. There are several AAV vectors which are being explored, but we do have 2 products which are currently in use. Luxturna is a locally delivered adenoviral vector which delivers genes subretinally. So you have you subretinally inject your genetic material using this adeno-associated viral vector, AAV2, straight away into the retina, and basically expresses and gives you the therapeutic response and improves the vision. Okay, that's basically brief overview of that. I've tried to talk a lot about different vectors, modalities. I want to briefly tell you about the limitations. We could have several limitations. We may, even though we want stable gene transfer in long term, we may not actually get that. We really need to think about that as well. We can have, body, even though we have made a lot of developments, body will still show immune response. It is still difficult to come up with a very innocuous vector. Especially viral vectors, as shown previously, they themselves can cause unwanted manifestation of the disease. They can affect a different cell, and that can lead to mutagenesis. When it comes to non-viral vectors, your transfection, they're not as powerful as viruses. So efficiency of gene delivery is something that needs to be considered. Whenever you have whenever you have disorders which have multiple genes involved, and if you were to treat those sort of diseases using multiple genes, obviously gene therapy is still not yet powerful to modulate multiple genes. Other people have tried that. And last but not the least is the cost. It's very powerful tool, we still have to consider the cost. Right? I mean, it's if you are able to afford that, only then gene therapy works. Okay. Well, just to conclude, I want to just tell you that gene therapy is a reality. I've tried to show you viral and non-viral vectors have both been successful in gene therapy. We've not really, although we had a lot of promise, where polymer-mediated gene therapy has not really been approved yet, so we still need to do a lot of work there. And currently, mRNA vaccines are really, really powerful tool for our infectious diseases, including COVID-19. All right, so that's all. I know it's a pretty long presentation, and I apologize for exceeding the time, but I really want to give you all an overview of that. So with that, I just want to say thank you, and I'm happy to take any questions that people may have, and we go from there.
Thank you very much, sir, for a wonderful session. Hello, everyone. So we had a very wonderful session by Dr. Date, and he explained many things, especially about the gene therapy, starting from why do we need gene therapy, then different forms of the gene therapy. Then later on, he also explained the barriers for gene therapy, like intracellular barriers for the gene therapy. Very importantly, he explained the different modalities in the gene therapy, and even also included the recently approved ASO and its development timeline. Then he explained about the small interfering RNA, then even the mRNA vaccine. Very important point, what he explained, the requirement for an ideal vector, then the vector for the gene therapy, even the fabrication of the LNP using the microfluidics. At the end of his session, he also explained the various new development in the gene therapy, even with some cases or with some research. He explained in very lucid language. And at last, he explained what are the limitations, including the cost factor for the gene therapy. Thank you, sir. Thank you for a wonderful session. And I would like to request Dr. Madhur ma'am for having the questions.
Thanks a lot, Dr. Abhijit. I know that the topic that you are covering is very, very broad. But then I'm very, very sure, and I'm sure all the viewers also would admit this, that you have really made a proper justice to the topic by covering like all the modalities of gene delivery. So the thing is, like, again, like, our chat box is flooded with lot of questions. The first one would be, see, you did mention about mRNA based vaccines. So the thing is, the first question is whether these vaccines, would they actually produce tolerogenic response? That's because, see, your own body is producing the antigenic material, and it's very likely that there would be a tolerogenic response, because there are no adjuvants. Like, usually all the vaccine deliveries incorporate adjuvants to get a proper immunogenic response. But here, your own body is producing, with the help of mRNA, it's producing the antigen. So would there be a chance of tolerogenic response?
Well, I think so. I think that's an interesting question. I mean, that's the other thing. That's one thing that certainly the field needs to address. I mean, like, after all, these are designed in a different way. One of the reasons that I personally think that it may actually really elicit is that if you are trying to modulate, I mean, when you try to exogenously deliver your immunogen, right? So I think that's a very different response using a vehicle, and you certainly need activation of immune system so that you can really get that immune response that you need, right? But when you're looking at delivering it inside and generating your immunogen at a really cellular level, right, and allowing your body to react to that, I believe, I mean, this is what I believe, that the response is certainly going to be pretty robust, and that's why you see a lot of success in non-human primates or even in clinical trials right now. I mean, whether it really relates to a stable long-term immune response or whether it's going to be just like, I mean, whether it's going to be a short-lived, which will have to be given repeatedly, I think that the time will only tell, right? So I think, yes, I think it's a valid point. I mean, but we are using lipid nanoparticles, right? So I don't see a reason why we can't include even, I mean, exogenously, we can include adjuvants so that we can certainly amplify the response that we get once we deliver mRNA.
Yeah, okay, thank you. There's one more question. What kind of sophisticated instruments and the lab setup that is needed for working on gene delivery? I mean, any kind of gene delivery.
Well, I think I mean, if you were to, again, I'll just talk briefly from lab perspective. So if you were to do some sort of gene delivery in the lab, I think you definitely need a cell culture facility because, you know, even for your simple in vitro transfection studies, you're going to require a decent cell culture facility. You definitely, well, I mean, like a lot of your sequences, I mean, we have companies, your IDT here or Thermocon, you know, you can actually buy nucleic acid sequences. You're not going to really have to worry about synthesizing nucleic acid sequences. You can actually design nucleic acid sequences. You can order them. You can even get a modified nucleic acid from Sigma or, you know, other IDT or other places. So I think you can certainly, if you have the money, you can certainly buy that. Sophisticated, I mean, yeah, certainly a couple of things to think about. You know, it's the nucleases, right? They're everywhere, right? So I think the practice is very important. You need to think about taking care of those so that you don't necessarily mess up your studies. I think for lipid nanoparticles, you can still use standard ethanol injection techniques if you have a mini extruder. That is relatively cheaply available, you know, simple injection-based extruder. You can certainly think about using that, or if you have a cationic polymer that you procure. I mean, sophisticated instrument-wise, I would say definitely your your cell culture hood. Whenever it comes to reading your output, I mean, gene silencing or siRNA silencing, you need a luminometer. So you need a very good plate reader with different capabilities so that you can read out, and you need to be able to have GFP sequences and stuff like that. I mean, I think the list can be really long. At the same time, if you ask me, can I not do it if I have cell culture facility and some money, obviously, to buy nucleic acid? I think I can still try it. I mean, it's not that impossible. Definitely the money, the sequence is something that you need to be careful about. I mean, I don't know in India whether we have places where other than IDT or a Sigma or other who can offer these sequences at a lower cost. I think that would definitely be a concern. I definitely agree with that.
Yeah. Okay,
Yeah,
thanks a lot, Dr. Abhijit. Over to Dr. Digam.
Yeah. So the next question is, can we use the gene therapy for type 1 diabetes, the type 1 DM? And if yes, then what way we can use it? Is there any published research or some kind of studies are going on?
Well, so I think that's an interesting question. Type 1 diabetes. I mean, yes, people have looked at. So essentially, for insulin, I think it's a different way. I mean, we certainly have genes. We certainly have theoretical means of figuring out a way to deliver that. But if I'm right, I think those studies, even though people have tried it, if I remember right, those studies haven't really gone too far when it comes to treating type 1 diabetes using genetic manipulation, right? I think it's a very solid idea. It makes total sense. We have the map, but from what I know or from what I've read, I don't think they've gone too far ahead. Certainly not up to clinical trials for those so that we can really treat type 1 diabetes.
Thank you, sir. One more question is there from the participants that can we use any other virus vector when we are having gene therapy, or we have to use a specific one?
Well, so I think, you have to be careful about with viruses. Or I know I didn't get a chance a lot to talk about viral vectors because that was not the topic of the discussion. But you have to understand that you need to have viruses which are not going to cause the problems of their own, right? So adeno-associated viral vectors are more popular because they are very less immunogenic. They can't infect the humans on their own. They actually need, in reality, they need other viruses so that they can infect humans, right? So that's why those are certainly much more preferred, and that's why we have products based on AAV. I mean, people have looked at HSV, people have looked at retroviruses of different kinds. Lentiviral vectors and lentiviruses and AAV are the ones that are popularly used. People use viral sequences for amplifying mRNA. So I think that's another way in which other viral sequences can be integrated into nucleic acid therapeutics, if not the entire virus. But I think people have tried so many different types, but I would say so far AAV and lentiviral vectors are the ones that are most preferred.
Yeah, thank you, sir. Thank you. One more question is there. Just like the oral insulin will be close to reality in coming future. So what do you think about oral gene therapy? So is it possible, or any group of scientists is working on it? Can we have the oral gene therapy in coming future?
Well, that's a good question. I mean, see, I think certainly people have been trying for that, right? I mean, like if not systemic administration of oral, even looking at locally delivered in the gut, right? I mean, it's certainly something that people have been looking at. I think, the barriers are even higher when it comes to oral delivery, right? I think one of the things that we have to understand is that even though we have a solid gene delivery, like a RNA-based product for intravenous administration, you have to think about it's mostly successful because we're targeting the liver, right? So far, we've not really been able to, unless you're going for a local delivery, we've not really been able to overcome all the other barriers when it comes to having systemically delivered nucleic acid therapeutic and target a particular organ. Now, when you think about oral delivery, you're adding so many additional barriers, the changes in the pH, just by passing through the gut, as well as the very harsh environment of the GI tree, right? While I would love to see availability of oral delivered siRNA or nucleic acid construct, I think it's still far away, but I would still hope to see that happening sometime down the line. Right now, I don't think I've seen solid effort. There are definitely effort. I don't think I've seen anything that's gone all the way up to phase 1 at least.
Hello. Yeah, I mean, this question, Abhijit, was by your mentor, Dr. Vandana Patravale, actually.
Okay.
I mean, this was the question which was by Dr. Vandana Patravale. So I'll just take up the next question that one of the participants is asking that you've been saying that lipid-based delivery so far has been quite successful among all the non-viral vector-based delivery of for the genes. But then the lipids could be, I mean, would you be able to achieve targeting with the help of lipids? Because there are many cells in the body which have these lipid receptors. So very likely that the gene would go to the wrong cell.
Well, I mean, so that's the thing, right? I mean, yes, I think certainly depending upon the lipid sequence or lipid chemical structure, and there could be a possibility, but you also have to think about the optimization. That's the optimization study, right? So that's why you are able to, especially with the MC3 lipid, you are able to get mostly apolipoprotein, right? So I think when you modulate the structure, yes, there is going to be extra off-target effects, even with lipid nanoparticles. I mean, they are far from perfect. I mean, there is no doubt that we need to do a lot of work. But if you'll see, mostly the approved products are either locally delivered, right? So you're going all the way down to the retina, right? Subretinal injections, right?
Yeah.
Or intravitreal injections. And the IV, yes, we definitely have IV products or even subcutaneous products now based on only self-delivering siRNAs. But you certainly could have that issue, and that's where a lot of optimization starts. I mean, Alnylam, basically their clinical trials initially failed, and everybody thought that LNP for siRNA was was gone, but you can see that even when you change the linker molecule, you can see the dramatic difference in siRNA delivery as well as overall targeting. Right? So I think all those optimizations would definitely have to be considered if you are developing other types of molecules.
Yeah, I think it's an extensive work. I'm sure an extensive research is needed into this area, because to be able to be successful, right, in delivering the gene to the right target. The other question that I had was, when you say that you complex the nucleic acids with the lipids or with the cationic polymers, the obvious question that comes to my mind, that if once it gets delivered into the cytoplasm, how the complex is going to break to set the nucleic acids free to do their action.
Right. That's a good question, and that's where I try to talk about the importance of pKa, right? So I think that's where the pKa design comes in, is very important, right? So what do you want, especially with cationic lipids? What do you want? Well, you do want cationic charge, right? So that they can bind to the negatively charged nucleic acid therapeutics and form that complex, right? So when it comes to formulation, you're using acidic buffer so that based on the pKa, your nucleic acid, your cationic lipid is highly positively charged, and you get that complexation first when you're building the formulation, right? Now, that's number one, where you now you have a stable formulation. Once it comes to, you definitely want a pKa in such a way that when you inject the siRNA containing lipid nanoparticle, that cationic charge, which is definitely going to be based on the pKa, does not cause toxicity, because if you have highly cationic molecules, they're going to bind to proteins, they're going to aggregate, they're going to do immune response, they're going to cause toxicity. That's very well known about cationic structures, right? So you don't want that, right? So when it comes to, when you have pKa somewhere around 6.5, when it comes into the body, essentially because of the change in the pH, your cationic lipid now adopts the structure, fairly neutral structure, so that it doesn't necessarily interact so much with the other proteins and doesn't cause a lot of toxicity or immune response, right? Now, you also wanted to go inside and deliver your cargo, right, which was your earlier question. So which is what I'm trying to get back at, right? So you can see you want sort of reversal of charges, right? And that's where the pK is very important. Initially, you want lower pH so that you can bind properly. You want that to change when you inject, because you don't want toxicity. Now, once it gets successful, it gets taken up by the cells and gets delivered into the cells. Right now, certainly it's going to be in a neutral state, but this is where your endosomes basically come into picture. And that's one major constraint when it comes to delivering nucleic acid therapeutics, right? Your endosomes are inherently designed to trap things, process kill your bacteria. They have acidic pH, and people take advantage of that acidic pH when it comes to cationic lipids. So now your endosomal pH is actually way lower than the pKa of your lipids that you've designed. Once your lipid construct gets inside your endosomes, basically the pH is lower. They immediately get positive again. And now they'll start fusing with the anionic lipids from your endosomes. And this is how the fusion, and that fusion will trigger the disassembly of particles and will release the siRNA into cytoplasm. So there is a lot that goes into the lipid design. And it's you can see the iterations of the lipids that people have come up with to figure out the right lipid which can successfully deliver siRNA construct or other nucleic acid construct in vivo.
Right. And then would you comment a bit on the proton sponge effect? Because proton sponge effect is one of the mechanisms on which we rely for the endosomal escape of the nucleic acids, right?
Right. So certainly, with PIs, you definitely PIs basically, which people have tried in the clinic too. They certainly rely on mechanisms such as proton sponge effect. The idea is that, well, you basically, if I were to simplify that, once they are taken up by the cells, you modulate the exchange of the protons, and you basically modulate the intracellular proton concentration as well as the osmotic pressure, and that itself will disrupt your endosomes and will release your cargo inside your cytoplasm and will lead to disassembly, right? So I think proton sponge is like highly cationic polymer. PEI is a classic proton sponge effect, a polymer that leads to proton sponge effect. So people certainly rely on that. But then when it comes to lipid, I think you can't, you can see that the charge balance is very tricky there, right? You can't have too high negative cationic charge as well. So it's very tricky. And part of the reason I believe, polymers are very efficient, no doubt, when it comes to in vitro. I think it's very difficult when it comes to managing them in vivo, right? Even though you have very, very cool mechanisms by which you get endolysosomal escape, it still is very difficult to, deliver things with that.
Yeah, that's true. And then, though PEI is very, very, very powerful cationic polymer, it has its own drawbacks, right? Like toxicity.
Oh, yeah, certainly. People have looked at pegylated PEIs. People have looked at ways to change that or mitigate the toxicity. But PEI in general, even though it's a very powerful when it comes to in vitro transfection, or it's not been very successful in vivo. People have tried that.
Right. And then with respect to the vectors, non-viral vectors, there's one question from one of the participants that can resealed erythrocytes be used as carriers for gene?
Well, that's a good question. I can't imagine. Well, I think that's an interesting question. It's an interesting thought for sure. I would still have, you're still talking about resealing. The intracellular milieu is something that you need to think about, right? When it comes to using, people have used cells to deliver genetic material, right? So I wouldn't necessarily say that you can't use, those as carriers. But I think you also need to think about that they're going to get extravasated everywhere, right? So I think one of the downsides that I can think of offhand, off the top of my head, is that the distribution can be good or distribution can be totally unwanted, right? You may be getting you may need a lot more genetic material because RBCs are going to go everywhere.
Right. It would be very haphazard. It would just get distributed everywhere.
Exactly, right? So it could be a good thing or it could be a totally horrible thing that can happen with the construct, if I were to just think off the top of my head.
Yeah. Thanks for that. Now, Dr. Digambar will just ask a few questions to you. Yeah, over to Dr. Digambar. Is he around? Okay, otherwise I will carry it forward.
Ma'am, please continue.
There's one question from one of the participants: how to control the duration of expression of gene after its delivery?
Well, I think so. I think that's, I'm not a geneticist, but I think that's got to do with the construct, right? You have to, your construct as well as your vector, both together will determine how efficient your gene transfer is going to be, right? And whether you're going to be able to get to the target and whether you're going to be able to, vector is certainly very, very important too, because, you really want that efficient delivery, but you also need to look at the genetic construct, which can essentially elicit that effect, right? So you need to think about both the aspects.
Right. And I think this could be a last question of this session. What are the possible adverse effects of gene therapy? Using non-viral vectors. Because we know viral vectors can cause immunogenicity and, it can have its own drawbacks. But then with non-viral vectors, what could be the adverse effects with gene therapy?
Well, I think, one of the limitations that we still see even with the non-viral vectors is still the immune response from the body, right? After all, these are synthetic lipids, right? Even though we have linoleic acid, we have all those things. And as I showed earlier, I think one of the reasons that Moderna has published a really nice paper that I talked about, right? So MC3, which is a very successful lipid, when you change the route of administration, clearly MC3 is not good for intramuscular, right? So I think because it sticks around longer, that material itself can lead to adverse response, right? So yes, I think even in the clinical trials, although broadly they've been accepted they've been acceptable by the patients. You do see side effects with non-viral vectors too, and part of them could definitely be associated with the materials, because after all, these are synthetic materials.
Yeah.
Even though based on lipidic materials which are partly endogenous, they will still definitely have some level of immune response, right? And the whole idea is that you want to minimize that, you want to minimize the toxicity, because you are still trying to deliver cationic lipids and as well lipids, right?
Yeah, right. And then I think with that, repetitive injections would be the most difficult task, because, like, say, if you delivering it once, probably the immunogenic response would not trigger that early. But if we go for repetitive delivery of the gene, I think that can lead to trouble, right?
Oh yeah, certainly. I mean, even with if I remember right, patisiran, you need, or at least in the Apollo trial, they were like doing once every 3 weeks, right? So I think that is still something that you need to consider when it comes to, see, at the end of the day, even with the gene therapy, right? It's a cost to benefit, risk to benefit, right? I mean, if you have a genetic disease for which there is no other treatment, if you have that, right? So you would certainly try to figure out a way. I mean, we worked with a collaborator on a rare disease called as pseudoxanthum elasticum, which is a calcification disorder, right? So where your calcification in your soft tissues gets messed up, and there's literally no drug available for that, right? So I think even though, yes, you are looking at some of the issues, but then if you are making these therapies available, I think that's the other side, right? So, I know it still needs to evolve. I mean, we are still not the end of the road. I mean, this is just the beginning.
Right. Yeah, I know. I understand. And just the last one again. I just said that we could take a last one, but then there's still some more questions coming up. Would sialic acid-based carriers be useful, or have they been tried?
Well, so I think, sialic acid, something similar to that, right? So I think I told you guys, polysialic acid, I think I saw that polysialic acid can be, tricky, because, you also have to think about the charge. It's certainly based on the endogenous ligand, but people have certainly taken cues from that, right? So the geosirin, which actually has N-acetyl galactosamine, which is sort of similar to, just target liver, right? So it's certainly going to help you there. So people have certainly taken cues from that. But I mean, I know that sialic acid-based modalities, people have looked at viruses, as an antiviral, or certainly as carriers. But, in theory, they wouldn't necessarily have that much. I mean, yes, you can build a construct on that. But when it comes to translation, I don't think polysialic acid per se could be that powerful. But yes, they have been successful. I mean, N-acetyl galactosamine, after all, is mimicking similar to sialic acid, right, which will allow you for targeting. So people have really taken cues from that.
Yeah, right. So, Abhijit, it was really an extensive session, and I know the kind of material that you have put in and you have really compiled. There are many of the participants who are actually asking for your slides, because you've put in such a fantastic information, and that too you've covered almost all the modalities of gene delivery. So it's just to ask you the permission whether you could share your presentation with our participants.
I think it should be fine. I think it's nothing different than, I wish we had a lot of our own. I mean, we are working on a few things, but we don't have enough data to talk about. But even otherwise, I have no problem. Even if we had a data of our own, that would have been nice. But I mean, this is all in public domain, right? So that's why I try to put links everywhere.
Right.
If you want to actually go and look at those papers, so that you can talk. I mean, people teach this topic a semester long or even more, right? So I think
I know.
I've been struggling with this presentation personally, just to how to, consolidate and try to give still a decent overview of all the things, right? So, and that's part of the reason why I ended up also overshooting the time. But I'm okay. I'm perfectly fine sharing the slides. I think it's totally okay.
Thanks a lot for that. And now I hand over the session to Dr. Kannur.
Yeah, very, very good afternoon, everyone. It has been a mind-blowing, fantastic session, Dr. Dade. We had a great learning experience, and the very way in which you have explained the nitty-gritties of the topic is simply awesome. I don't feel any other person could have simplified it to this extent. And all the comments and even including all of us, it is very much fantastically done by you. We are extremely thankful to you, heartfelt thanks. Yeah. And you are a part of team ICP, so it is a great honor again to have you on this session. And we look forward again to have many more sessions of yours and have a long term association with you. And we are also thankful
Of
to
course.
all our eminent participants who have who join at a very what do you say, a particular time, and everyone is there online, and active discussion is there. So this is a great platform and a great learning thing. We are also thankful to Dr. Madhur and Dr. Digambar for the moderation part also. So on part of Team ICP, I express my sincere most thanks to Dr. Date as well as all the eminent participants. Thank you very much. A small announcement for tomorrow. Tomorrow's session timing has been changed slightly. Instead of morning 10:30, the session will be conducted at 4 p.m. in the evening. So you all are kindly requested to join as per the daily procedure, say by 3:55 p.m. and we will be starting the session sharp at 4 p.m. So thank you very much. Dr. Ratnesh Kumar will be addressing us tomorrow, and we eagerly look forward to have you again tomorrow. And once again, a heartfelt thanks, Dr. Date. Thank you very much.
Well, thank you so much. I hope
Thank
I
you,
did
sir.
this. I really appreciate
Thanks.
that. And well,
Thanks
friends,
a
Thanks a
lot.
lot, Abhijit.
love to be a part of it. Yeah. Thank you.
Yeah.
Take
Thanks.
care.
Thank
Thanks a
you.
lot.
Well,
Yeah, bye. Bye.
And
yeah,
bye.
bye.
Bye,
Bye
sir.
bye.
Thank you.
Bye.